The advances in gene editing are a game changer. Our Pathfinder Award goes to Dr. Feng Zhang. Feng is a molecular biologist who has developed novel molecular techniques for studying the brain. He pioneered the development of CRISPR-Cas9 as a genome editing tool and has substantially expanded the use of CRISPR tools. One day, CRISPR might be harnessed to treat and possibly cure debilitating medical conditions such as sickle cell anemia, Duchenne muscular dystrophy, Huntington's disease, HIV, and cancers. Feng is currently a core institute member of the Broad Institute of MIT and Harvard, as well as an investigator at the McGovern Institute for Brain Research at MIT. We have two individuals, advocates, that will describe the importance of Dr. Zhang's research. Both have been honored by the Regenerative Medicine Foundation. In 2012, we honored Susan Solomon and the New York Stem Cell Foundation. Susan is now the founder and the chief executive officer of the New York Stem Cell Foundation Research Institute, which is the world's leading independent nonprofit research institute dedicated to translating cutting-edge stem cell research into clinical breakthroughs and cures for patients. Located in Manhattan, it is absolutely an astonishing feat of advocacy that foundation has created and is doing independent, world-class research. Also, we're going to hear from Frances Saldana. Frances and her family has been afflicted terribly by Huntington's disease. We honored the HD grassroots community with an inspiration award at the World Stem Cell Summit in San Antonio in 2014. I had met Frances in 2011 in Pasadena at the summit, and it was there I learned about the affliction of Huntington's disease and the devastating toll it takes on those that perish and those left behind to deal with this. Gene editing provides hope.
I'm Susan Solomon, the CEO of the New York Stem Cell Foundation Research Institute, and it is he does not want to be on one side of this divide or the other. He wants to show us how wrong and unnecessary this split really is in the first place. In this sense, Feng represents the very best of science. He goes where his curiosity takes him, and his curiosity is a magnificent engine driving his brilliant work, pushing him and his laboratory in dozens of projects simultaneously. Last year, when the severity of the pandemic became clear, Feng immediately pivoted his technologies using the groundbreaking CRISPR gene editing to develop a faster, cheaper test for COVID-19. And by May of 2020, he had a prototype that cost $6 per test. He also developed a self-reporting app called How We Feel that tracks changes in the spread of COVID-19. We at the New York Stem Cell Foundation Research Institute have been privileged to have known Feng for most of his career. He was one of our early Naisif Robertson innovators And when his 5-year fellowship ended, he became one of our most engaged alumni. His mentorship and involvement with our new grantees, events, and scientific retreats have set the standard for community building. His novel approaches and enthusiasm remain the high bar for entrepreneurship at the highest levels. He communicates about science with the clarity of a natural teacher, and he always answers emails a few hours after I send them, despite the demands of his lab and the young family to whom he is a devoted father. Walter Isaacson, in his recent best-selling book about gene editing, describes Feng multiple times with the word gracious. And Feng is all that this word implies: caring, engaged, committed, knowing, and kind. He is truly in a category all his own. So Feng, I speak on behalf of all of us here when I say that it is an honor and a privilege to recognize your significant contributions with the Pathfinder Award. I'm so happy to call you a colleague and a friend. Congratulations.
Hi, Dr. Zhang. Congratulations on being named the recipient of this year's Distinguished Stem Cell and Regenerative Medicine Action Award. Your brilliant work as a pioneer in molecular biology and your hope of improving human health could not be more meaningful to me. This is because I have lost my mother-in-law, the father of my children, and all 3 of my children to Huntington's disease. After having lived through 3 generations of genetic diseases, the story doesn't end there, and neither does my hope for a cure. The vital role that you have played in the development of CRISPR-Cas systems may one day lead to a treatment that will save so many lives throughout the world. Like my own family, these are families who are anxiously awaiting a treatment that will free them from a disease that is passed on from one generation to the next. A world free of Huntington's disease in my own lifetime would be my greatest wish. My children never lost hope for a cure, and your continued search for treatments for neurological diseases such as HD allows me to keep their spirit of hope alive. So on behalf of Huntington's disease families from all over the world, I feel honored for the opportunity to thank you and to congratulate you for your brilliant work. Thank you and congratulations.
I would like to thank the Regenerative Medicine Foundation for this tremendous honor. This recognition is not just for me, but for my entire team, my collaborators, mentors, teachers, my parents, and my family. I want to take this opportunity to say a couple things about teachers and mentors, especially during primary and secondary education. I was very fortunate to move to the United States 28 years ago with my parents, and I'm grateful to all of the teachers who have inspired and supported my learning and development. From my English as a second language teacher, Marilyn Ritz, who taught me English and American culture, to my science teacher, Ed Pilkington, who showed me the movie Jurassic Park and inspired me with exciting advances in biology through simple but memorable experiences, such as extracting DNA from strawberries. And my first research mentor, Dr. John Levy at Iowa Human Gene Therapy Research Institute, who kept me captivated with his passion for gene therapy and the destruction of cancer cells. And also my graduate school mentor, Carl Deiserath, who not only taught me how to be a scientist, but also how to be a catalytic member of the scientific community through open sharing of knowledge and technologies. There are numerous other mentors and colleagues I am indebted to, and that list is too long for me to recite today. But most importantly, I'm grateful to my parents, my wife, for their mentorship and support. They have instilled in me the value of striving for excellence and generosity. They have always emphasized to me the importance to always do my best and to create things that can make the world a better place. And that has been a guiding principle for me all along. Finally, I want to say how exciting it is to be a biotechnologist today. Our understanding of biology is growing at an ever-increasing rate, and our capabilities for illuminating biological processes and turning those understandings into new therapeutics is unprecedented. I'm incredibly inspired by this enormous opportunity, and I am looking forward to working with my team and continuing to collaborate with researchers around the world to advance human health.